Our speaker today. Dr. Manuel Gahete completed his postdoctoral training here at UIC in the laboratory of Dr. Kinneman, and then he returned with a prestigious postdoctoral fellowship to Spain. Since 2021, he's associate professor with tenure in the Department of Cell Biology in the University of Córdoba in Spain, and he's the head of the molecular hepatology group in the Maimonides Biomedical Research Institute of Córdoba. Dr. Gahete is an excellent scientist that has published over 130 publications in the field of endocrinology, metabolism, and translational oncology. And he is the principal investigator. He has been and is the principal investigator in more than 11 research projects funded by extramural agencies. This is not only the thing, is he has received multiple prestigious international awards, including the Young Investigator Award and the Jens Sandal Christensen Award of the European Society of Endocrinology, the Young Endocrinologist Award from the Spanish Society of Endocrinology, the Early Investigator Award of the Endocrine Society, and the Rising Star Award of the Spanish Hepatology Association this year. So congratulations, Manuel, on those recognitions, well deserved. And it is an honor to host you today as our speaker. And thank you for sharing your time and your result with us. And the Zoom is yours.
Okay, thank you so much, Jose. I'm gonna try to share my screen. If you can check that everything is working. I cannot hear you.
We are getting again that bar on top.
On top? That's, I don't know why, because I did just exactly the same that the last time, but let me. And now?
Now, yeah, we've seen it, but we see the title of the slide.
Okay, perfect.
You can go.
Thank okay, you. So thank you so much, Jose, for the kind, more than kind introduction, for the invitation to be here. Thank you, and also the Department of Medicine and the UIC for hosting me today. For me, it's a great pleasure, but also an honor and also a responsibility to be here delivering this talk, because as you mentioned, I was doing my postdoc there with Dr. Rhonda Kinneman. It was an excellent time to be there, and now I have the responsibility to share what we are doing right now here in Córdoba when I came back from this postdoc. So I hope you enjoyed the talk, and we can discuss any question at the end of the talk if you want. As Jose mentioned, I'm associate professor at the University of Córdoba and also principal investigator of the recently created group of molecular pathology. We are a small group in the Biomedical Research Institute, but we are working with the general aim of trying to understand some of the cellular and molecular mechanisms that underlie the development and progression of liver diseases in general. We are doing that because, as you probably know, chronic liver diseases, or the different stages of the chronic disease, are a rising problem for the health and for the health systems also. I'm not going to talk in detail about all the steps of the chronic liver disease progression and all the data, but I think there are some important data that I would like to share and remind you. In general, MAFLD, or non-alcoholic liver disease, is the main cause of liver dysfunction worldwide. The prevalence in the general population is around 20 and 30%. As you can imagine, the prevalence in the obese population is higher. It's between 60 and 70%, depending of the study. As you probably know, it has been proposed to be renamed as MAFLD, what means metabolic associated or metabolic dysregulation associated fatty liver disease, because it has been found that this MAFLD or NAFLD is clearly associated with metabolic syndrome, with obesity, with diabetes. This term, NAFLD or MAFLD, is like a word that is trying to incorporate a spectrum of chronic liver diseases, where we are ranking from simple steatosis, which is the ectopic accumulation of lipids in the liver. This is kind of a benign pathology where we don't have too many problems. It's if we only have simple steatosis, we have fatty liver, but this is kind of a reversible situation with diet or exercise or even with surgery. So, but the problem is that this simple steatosis can progress to more chronic stages, such as non-alcoholic steatohepatitis or NASH, which include the development of liver damage, ballooning, inflammation, can be also associated with fibrosis. NAFLD and NASH are, as I mentioned, kind of reversible stages that you can prevent, or using diet or exercise, we can also kind of partially revert. But the problem is when the pathology go further and we start to have cirrhosis in the patients, because cirrhosis is clearly associated with the development of hepatocellular carcinoma. And we know that the hepatocellular carcinoma is the main primary liver cancer and is associated with the poor survival of the patients. Depending on the study, the survival of the patient is ranging between 10 and 30% after 5 years, which is a real problem for the patients and for the health systems. But this classic progression of the chronic liver disease is being changed with the recent studies that are suggesting that it's not so linear always, but NASH can directly progress to hepatocellular carcinoma, and there is also some studies demonstrating that even NAFLD or the presence, we don't need the presence of cirrhosis or fibrosis to develop hepatocellular carcinoma. So there is much complex that initially envisioned, probably. And this is also a problem because we know that all these liver pathologies are associated with the obesity. NAFLD is the main hepatic manifestation of metabolic syndrome, of obesity and diabetes, which are pathologies that are increasing in prevalence in the worldwide population. And this is doing that we need to change this is making that the global epidemiology of hepatocellular carcinoma is changing, because classically hepatocellular carcinoma was appearing in livers from patients with viral infection or with a high consumption of alcohol. But now we have a lot of cases where this hepatocellular carcinoma is developing in livers of patients with NAFLD or NASH. You don't need to have a viral infection or a high alcohol consumption to develop hepatocarcinoma. And since obesity is increasing in prevalence, NAFLD is also increasing, and hepatocellular carcinoma is also increasing. Indeed, now liver cancer or hepatocellular carcinoma is the third most prevalent cause or cancer-associated death in the world population. That's why we are starting to study these different steps of this pathology. We are developing different projects to trying to understand which is the progression of the disease, which are the different stages, and how we can modulate this progression. We have been obtaining funds from different agencies to study different steps of this pathology. The final aim of those projects is to try to identify potential biomarkers or even novel therapeutic targets to improve the management of the patients with some of these chronic liver diseases. To do that, we are applying different approaches. We are using high throughput approaches to trying to understand or try to gain more insights about the general dysregulation of the cellular and molecular mechanisms in these pathologies, including quantitative proteomics or RNA-seq, but we are also using more focused, more target approaches using a methodology that we have been developing and optimizing in the lab. I will talk about it later, but it's a microfluidic-based qPCR technology, but usually using also some kind of usual commercial arrays to measure mRNAs, phosphoproteins, and all kind of commercially available kits. And we are also using classic molecular biology approaches to confirm or to validate or to demonstrate those changes that we are observing with the more high throughput or medium high throughput approaches, like the classical qPCR, immunohistochemistry, Western blot, etc. The idea is to try to characterize which are the changes, at gene expression level and at protein levels of different components that we can eventually use as novel biomarkers or therapeutic targets in these pathologies. And after we identified these elements, we use a series of additional functional and mechanistical studies to try to understand which is the implication of these changes that we have observed in these pathologies, which is the implication in the real development of progression of the pathology. To do that, we are using cell lines and primary cell cultures where we can modulate gene expression using silencing or overexpression of this gene or protein or interest, or even pharmacological blockers or pharmacological activators to modulate the activity of these proteins. And we are complementing those results, and here is where our collaboration with Chicago groups is more fruitful, is using the preclinical models. We are using xenograft models, but also transgenic or diet chemically induced animals to confirm what we are observing in vivo. As I mentioned, we are, or as you could see in the title of the talk, we are especially interested in exploring which are the dysregulation that are occurring in all the steps that are controlling gene expression, because we know that during the development of the cells, or during the normal physiology, or even during the development of different pathologies, gene expression is changing. And gene expression is a highly complex and obviously regulated process that can be controlled at different levels. It is obvious that there is a genetic and epigenetic modulation of gene expression. There is some modification in the DNA, some methylations and epigenetic modulation that are going to define which genes are going to be expressed and which genes are going to be repressed. But then after one gene is started to be expressed, there are also a lot of these different mechanisms that are controlling which is the final product of this gene, which is the mature mRNA that is going to be produced. When a gene is starting to be expressed, what we generate is a pre-mRNA, and this pre-mRNA has to be maturated. There is some steps of maturation of this mRNA, including capping, polyadenylation, but also the splicing. Most of the mammalian genes are composed by different exons and introns that have to be combined in a different ways to produce the mature mRNA, and this is the splicing process. And this is crucial for the appropriate maturation of the mRNAs. And once these mRNAs are produced and generated, they have to be exported to the cytosol, and then there is different machineries we will see later that decide if this mRNA is appropriately maturated or not, and it could be degraded or finally translated to protein. And this is where we finally obtain the mature mRNA that is going to be generating a functional protein. We know we have every day more data indicating that there is a very complex process, a very complex machinery that is controlling all these steps of mRNA maturation. And this is critical for the appropriate functioning of the cells, because when these systems are altered, then we have a pathological progression of some diseases. And we are especially focused in exploring how these machineries that are controlling the different steps of maturation of the mRNAs are altered in different pathologies. And we are interested in that because we know that during the progression of the different diseases, and especially during cancer development, there is what is called a transcriptional reprogramming. We have a change in the landscape of mRNAs that are produced during the pathological progression of certain diseases, specifically cancer, and this is associated with the alteration of those mature mRNAs, which are associated with specific clinical, with specific cancer hallmarks. Indeed, there is now the concept of cancer transcriptome atlas that indicate that different tumors of different tissues have a specific transcriptome that is associated with clinical characteristics of the patients. In the case of hepatocellular carcinoma, there are some studies demonstrating that the specific landscape of mRNAs, what is called the transcriptome, is altered during the progression of the disease. And this can give us some clues about new therapeutic approaches, but it is also associated with the progression of the disease. In this study, it was demonstrated for the first time that different stages of the disease are associated with different changes in the transcriptome of the tumor. These also have clinical implications because the patients with a specific set of genes that are overexpressed or downregulated are going to be associated with certain clinical characteristics. When they did this analysis in this study, they observed that two of the main pathways associated with the clinical outcome of the patients was two pathways associated with gene expression, those that are in the red circle, the alternative splicing and the transcriptome. Again, demonstrating or suggesting that there is a dysregulation in this process of gene expression that is associated with tumor progression and with the clinical outcome of the patients. Indeed, there are a lot of different studies, not only in liver pathologies and not only in hepatocellular carcinoma, demonstrating that in cancer in general, there is a dysregulation of the alternative splicing process, which is one of the main processes controlling mRNA maturation in the cell. And this is associated with the generation of specific oncogenic splicing variants, which are associated with different cancer hallmarks, including proliferation, metastasis, angiogenesis, apoptosis. So this process is dysregulated during the progression of the disease and probably associated with the clinical outcome of the patient. The problem, and there is some studies demonstrating that this process is altered because we have the end point of this process that are these specific splicing variants. There are a lot of studies demonstrating that in certain pathologies, including hepatocellular carcinoma, there are new splicing variants that have oncogenic potential. The question is why those splicing variants or why those aberrant mRNAs are present in tumoral cells or in cells that are suffering some pathological process. The idea is that those oncogenic or pathogenic variants are there in the cells because there was a problem with the machineries that are controlling the maturation of these mRNAs, because in physiological conditions, in normal conditions, those variants should not be here or should not be there. So that's the idea beyond the exploration of which are the dysregulation of these machineries. When we are talking about cellular machineries controlling mRNA maturation process, obviously one of the main one is the spliceosome, is the machinery that is controlling splicing. But there are also a couple of them that are very important for the surveillance of those mRNAs that are trying to be mature, or those mRNA that are in the process of maturation, that are the RNA exosome and the nonsense mediated decay machinery. Those machineries are controlling that those mRNAs that are not appropriately maturated cannot be exported to the nuclei, to the cytoplasm, or cannot be translated to proteins. And if finally those pathological mRNAs are translated to proteins, it implying that these machineries are not appropriately doing their work. And this is the idea why we are exploring, which is the dysregulation of these cellular machineries in liver pathologies. One of the machineries where we have been working more profoundly during the last year is the spliceosome. The spliceosome is a very complex macromolecular machinery that is constituted by a catalytic core that is reading the mRNA and detecting where an intron is present to be eliminated, but also is associated with a set of multiple proteins that are helping this core unit to do its works. So finally, we have like 200 or 300 different non-coding mRNAs and proteins that are detecting where we have an exon, where we have an intron, which intron have to be eliminated, which exon have to be kept into a mature mRNA. The problem of studying this machinery is there is not commercial array of, there is no way to explore the full machinery at the same time. And that's the reason we have been working to generate and optimize this microfluidic based qPCR array, which is a kind of array based on microfluidic using a specific equipment that allows us to detect or determine or measure the expression of almost 100 genes or 100 transcripts at the same time in 100 samples. And we have been using this technology to characterize which is the pattern of dysregulation of the splicing machinery, but also other cellular machineries in different pathologies. We have been using this same technology to characterize the splicing machinery in diabetes, in MAFLD, and more recently also in hepatocellular carcinoma. So what I'm going to do is give you some examples of what we have been doing using this technology and how we implemented that in these different pathologies and which are the results that we have obtained. The first publication that we did using this technology was this paper that was published in eBiomedicine. It was a collaboration with Professor Lopez Miranda here in the University of Cordoba. Probably some of you know him because he has he is the lead of this study that is called the CardioPrep. Has been this study, the main result of this study have been published recently in Lancet. And this is a study where they recruited more than 1,000 patients that suffer a cardiovascular event before the inclusion into the study. They basically had a heart attack. They were included into the study because of that. The rationale of the study was to put those patients into diet in order to prevent a second cardiovascular attack or cardiovascular complication. When they were analyzing this cohort of patients, they realized that some of those patients had already diabetes, while, as you can see there, 462 patients did not have diabetes at the time of the inclusion in the study. They were following those almost 500 patients during more than 5 years, but they did a cutoff after 5 years, and they detected that after that time, 107 patients were developing diabetes at any time of the study, while 355 patients did not develop diabetes. What we were doing was taking samples from all those patients that developed diabetes, the 107 patients, and we selected also a cohort of 108 patients that did not develop diabetes. We took samples, blood samples from those patients. We isolated the peripheral blood mononuclear cells because we know that those cells are kind of representative of what is happening in other tissues because they are responding really nicely to metabolic changes that are occurring in the patients. And we took those cells to measure the expression levels of representative sets of components of the splicing machinery. What we found is that at basal level, at the time of the inclusion on the study, where all the patients were non-diabetic, we could observe already changes in the expression of some of these elements in those patients that were going to develop diabetes during the progression of the follow-up of the study. So we took patients that were going to develop diabetes, some patients that were not going to develop, and before the developing of the diabetes, they were already changes in the expression of the splicing machinery in those patients. Indeed, we were able to identify a specific molecular fingerprint that was capable to predict which patients were going to develop diabetes after 5 years of follow-up. And this predictive molecular fingerprint that was based on the expression of the splicing machinery was better than the classical predictors of diabetes development, such as fin risk or glycated hemoglobin. So there is the idea was that there is a dysregulation in the splicing machinery that is associated with the development of diabetes, and that these changes occur before the development of diabetes. We don't know the...
I'm sorry to interrupt, but I missed the part where you say how the samples are collected or...
Oh, yeah, sure. Yeah. What we did was we collected blood samples from the patient at the inclusion of the study. And at the inclusion of the study, they were non-diabetic still. Mm-hmm. So, but during the follow-up, during the 5 years of follow-up, some of them developed diabetes, while some of them did not.
Okay, and this is all gene expression in circulating cells then?
Exactly.
Okay, thank you. Sorry about that, yeah. Yeah.
So the idea was that in circulating cells of those patients, we could observe or we could detect changes that were associated with the development of diabetes in 2, 3, 4, or 5 years. We don't know exactly which is the link, but we did also an in vitro study where we treated circulating cells of normal patients I mean, normal patients were people from the lab, young people from the lab that were treated with the serum of those patients that were going to develop diabetes or not. And we could observe that the circulating cells of control people were responding to the serum of the patient in the same way that the changes that we were observing before the inclusion on the study. So the idea is there is something
you
in
know,
the
I just
serum.
went through a whole thing online.
Sorry?
You know, identifying things that I didn't think were correct.
Well, that's the idea. So if we treated,
Right. There's
if we
quite a
treated,
few. And
yeah.
Yeah, I think I know what happened. I responded to a text message
Hey,
that I
Terry,
thought was
Terry,
from the United States Postal
your
Service, but
Terry,
obviously
microphone is on.
it was a
Terry.
hack.
Let me
So I
see if
think
I
that's what
can
happened. And
mute.
Hey, Jose, I think you
I
can
did it. I
mute
muted.
See,
everybody.
you can mute him. Okay, thank you.
Sorry.
So, okay, that's the idea. So we don't. The idea is that should be something in those patients, in the serum of those patients, that is modulating the expression of these splicing components in the circulating level. So we could detect those changes and some way predict which patients were going to develop the diabetes or not. We don't know exactly which are the serum elements that were controlling that. Indeed, we were doing different approaches to trying to identify which one. We couldn't do that. But the idea, the final idea, is that the splicing machinery is changing in those patients that they were going to develop the diabetes, and we could some way predict which patient were going to do that. This, as I mentioned, this was the first study doing using this technology, but we use it also to study changes in MAFLD. That is one of the main focus of the group right now. We were doing a collaboration with a group of Dr. De La Mata, where they were collecting tissue samples, liver samples from obese women with or without steatosis, where they were characterized within steatotic or non-steatotic patients. They were all of them obese women, and we use the same technology to measure the expression of this splicing machinery in the liver of those patients. And we observed that the expression of certain components of the splicing machinery was clearly dysregulated. And again, we were able to identify a molecular fingerprint that could be discriminating between livers of patients with steatosis or patients without steatosis. And not only that, but if we forgot about those patients without steatosis and we just focus on the patients with steatosis, we found that not all of them have the same changes in the splicing machinery. When we did a unsupervised clustering of those patients, we could identify clearly 3 different groups of patients that were characterized or associated with specific changes in certain splicing machinery components. And they were not only characterized by specific changes, but they were all also associated with clinical changes. So patients that were characterized by changes in certain components of the splicing machinery had also certain clinical characteristics that were different to other patients. For example, patients included in the cluster A were characterized by higher glucose levels or higher aptoglobin levels, while patients included in cluster C were associated with higher insulin or higher ALT levels, indicating that depending on the changes that are happening on those patients, the clinical implications could be also different. to trying to understand if this association were cause or consequences, where we were using some in vitro approaches. We were treating HepG2 cells with lipids. We were inducing lipid accumulation on those cells, and we observed that the accumulation of lipids on the cells were not associated with changes in the splicing machinery, suggesting that the splicing machinery is not changes as a consequence of the lipid accumulation. However, when we did just the opposite approach, when we silenced or modulated the expression of these splicing components, then we could be able to observe changes in lipid accumulation, suggesting that the changes of the splicing machinery occur before the accumulation of lipid in the liver. This obviously opened a new avenue in the study of the group, and we are trying to characterize which are the changes in the splicing machinery that occur before, during the progression of the MAFLD, and which are the main changes, because obviously not all the elements are going to be changes at the same time, and not all of them are going to be important. The key thing here is identify which are the main drivers or the main changes that occur during the progression of this disease, that is MAFLD. But as I mentioned at the beginning, we are interested in the different steps of the chronic liver disease, and we have been studying also hepatocellular carcinoma. We have been able, through collaboration with the clinical groups here in the university, in the hospital, we have been able to obtain samples from different retrospective and prospective cohort of samples. We have samples from tumor and also from adjacent non-tumoral samples. And we have been studying the expression of these samples and comparing those pattern of expression with data that we can have available in the repositories elsewhere, in silico cohorts from different studies. And we have been using this retrospective cohorts and in silico cohorts to characterize not only the spliceosome, but also the other machineries that I mentioned before, the RNA exosome, the nonsense mediated decay, but also additional cellular regulatory system that could be implicated in the development and progression of hepatocellular carcinoma. But again, the main results are associated with splicing machinery. We were using the same technology in hepatocellular carcinoma, and we identify drastic changes in the splicing machinery in hepatocellular carcinoma samples. And we were comparing with what we obtained in our cohorts with what is there available in other in silico cohorts from different populations. And doing this study, we identify basically 3 spliceosome components that are commonly dysregulated in the same way in all the different cohorts that we have been investigating, that are SF3B1, A43, and also PF8, that are the ones that we have been exploring more in detail. The first one that we have been interested is SF3B1, because this is the core component of the splicing machinery in the cell. It is doing the first step of splicing during the splicing process. It is detecting where an intron is and cutting the sequence. And we have found that SF3B1 is overexpressed in hepatocellular carcinoma. This was not a surprise because it was reported to be overexpressed in different pathologies or different cancer types, but was clearly associated with the survival of the patients. Patients with high expression of SF3B1 in the tumor had lower survival than the rest. So we use some of the approaches that I indicated at the beginning. Basically, what we did here was to silence SF3B1 in 3 different cell lines of hepatocellular carcinoma, and this was associated with a reduction in cell viability, cell proliferation, and cell migration. One of the main characteristics of SF3B1 is that there is a lot of pharmacological inhibitors of this molecule, because it has been postulated as a therapeutic target in different pathologies. And in this case, we use a pharmacological inhibitor that is close, it is named pladinolide B, to try to reproduce the same results. And what we observe here is that if we treat those cells with the pladinolide B, we can reduce again proliferation, migration, tumorosphere, and colony number, indicating that this is a general inhibitor of the tumor progression. And a good thing of this inhibitor is when we treated normal-like cells, these THLA2 cells, the bars that are in orange, the inhibitory effect is much less pronounced than in tumoral cells. So open probably utility in the treatment of those patients. We demonstrated that also in vivo, not only in vitro, using xenograft mice. What we did was to induce the tumor with Hep3B cells in nude mice. And once the tumor was growing, once the tumor was already established, we injected pladinolide B, and the classical treatment of those patients, that is sorafenib. This is not quite exact right now, because there are new treatments in hepatocellular carcinoma, but at the time that we did the study, sorafenib was the gold standard. Pladinolide B is at least as potent as sorafenib in the blockade of tumor progression, and probably it has much less side effects because it's not a chemotherapy like sorafenib is. So this again is in vivo, demonstrating the same that we saw in vitro. The second of the study that we did was A43. This is also a very important component in the splicing process, because it's controlling different steps of the process. The good thing of A43 that was important for us is that was overexpressed in absolutely all the cohorts that we were exploring, absolutely in all of them. So this seems to be a very consistent marker in hepatocellular carcinoma. Indeed, I did not include the results here in the presentation, but we could detect A43 in plasma of the patient, and were also increased in patients with hepatocellular carcinoma. And again, the in vitro studies were indicating again the same. If we silence A43 in cell lines, we reduce proliferation, we reduce cell migration, we reduce colony number and tumorosphere size. But also the opposite is true. If we increase the expression of A43, we see absolutely the opposite phenotype. We increase proliferation, colony number, and tumorosphere size. And we have also an inhibitor for A43, and we use it obviously in vitro, and we confirm that the inhibition of A43 can reduce again proliferation, colony number, or tumorosphere size. In the case of A43, we had the possibility to study some in silico data, and we identified that we demonstrated that A43 is associated with the altered or aberrant splicing of different genes, including one that is very important for the progression of hepatocellular carcinoma, that is FGF4, FGFR receptor 4. FGF receptor 4 is the endogenous ligand for, the endogenous receptor for FGF19. And this signaling pathway is really important during liver cancer progression. We found that when we silence A43, we reduce the expression of FGF receptor 4 but also we modulate the expression of, we induce a shift in the mRNA species of FGF4 that are generated. When we silence A43, we induce the generation of FGF receptor 4 isoform that is not appropriately processed. It is lacking this signal peptide that is sending the receptor to the membrane. So if A43 is reduced, FGF receptor 4 cannot go to the membrane, and then FGF19 cannot signal as an oncogenic signal. And this is what we demonstrated here. If we silence A43, the cells are not responding to the oncogenic signaling of FGF19. Indeed, we demonstrated that doing rescue experiment. In this slide, you can see that we did at the same time the inhibition of A43, but at the same time the overexpression of a variant of the receptor that cannot be spliced. And in this case, we totally recover the signaling of FGF19 and the signaling of the receptor. So demonstrating that probably A43, this splicing variant, is inducing the oncogenic pathway, stabilizing controlling the appropriate process of this oncogenic receptor that is FGF receptor 4. I don't wanna be out of time, Jose. So, but if I can show the last paper, I will do it very quickly.
Okay, go ahead.
Is PRPF8 is the last one that we have published just like a couple of months ago. This is also an important component in the splicing. Is again overexpressing in hepatocellular carcinoma, also associated with the overall survival of the patient, and we did a similar approach. This is from my point of view a really nice element, because it is associated with the capacity of the cells of migrate and probably to do metastasis. We did a similar approach. When we reduce PRPF8, we can see a reduction in the capacity of migrate of the cells, the capacity to migrate, but also to form colonies. Also in animal models, if we silence PRPF8, we block tumor development. And this is associated with the splicing of a protein that is very important for the migration of the cells, that is fibronectin-1. Fibronectin-1 is a protein that is important in the extracellular matrix. Is the protein that is intracellular in the extracellular matrix, and that the tumoral cells use to induce the starting of the migration and the metastasis. We found that PRPF8 is essential for fibronectin-1 to do an appropriate splicing pattern. If PRPF8 is not present, the fibronectin-1 is going to lose a domain that is important for the integrins to be recognizing fibronectin-1. And if we, as you can see there, if you reduce PRPF8, we induce a shift in the two species of fibronectin-1 that can be produced. And in consequence, what we see is that if we reduce PRPF8, we have a reduction in the signaling induced through the AKT-PAC pathway. That is a pathway that is very important for the migration of the cells. As you can see here, the cells that are losing PRPF8 have lost the possibility to induce stress fiber formation. They cannot migrate, they cannot initiate the process. And we do the same with doing invasion assays. If we reduce PRPF8, we are reducing the capacity of those cells to migrate and induce the invasion of the adjacent tissues, which postulate PRPF8 as a good target to avoid the migration and probably the metastasis of those tumors. As I mentioned just a couple of minutes ago, we are not studying just spliceosome, but also other machineries, and we have been observing similar results. There is a strong dysregulation of the RNA exosome machinery, also associated with the survival of the patient and also associated with in vitro parameters, but also for the nonsense mediated decay, which is also dysregulating in hepatocellular carcinoma and associated with probably with the clinical parameter or clinical characteristic of the patients. So the idea that I wanted to share with you is that there is a profound alteration of the transcriptional landscape in liver pathologies, which could be probably associated with a dysregulation of the cellular machineries that are involved in the maturation of and metabolism of this mRNA. For that reason, we think that analyzing the alteration of these machineries, spliceosome, RNA exosome, nonsense mediated decay, can serve to identify novel biomarkers and therapeutic targets in this pathology. Our results suggest that alteration SF3B1, A4A3, PRPF8, and also additional elements are associated with the outcome of the patients and probably with the response to the treatment. These studies have also demonstrated that some specific pathologic splicing variants, in the case of FGF receptor 4 or fibronectin-1, are potential candidates to develop novel therapeutic strategies in liver diseases and hepatocellular carcinoma. And I don't want to finish without thanking, obviously, all the members of the team for doing this study possible, but also to the people of the team that is led by Raul Luque and by Raul Luque himself also, because we have been doing those studies in collaboration. And thank you, Jose, thank you, Rhonda, and thank you for all the people that is today here with me. Thank you so much.
Great presentation, Manuel. Amazing results. Thank you for sharing them with us. So we have time for some questions. I mean, if I could go ahead, I mean, I think it's amazing that you can get that blood from students. So that I remember a talk from a guy doing this cold exposure from Japan, and he said that also the students they were volunteering to do the cold exposure in the courtroom.
Yeah. They were absolutely volunteering. They were not forced to do that anyway.
Okay. So I think that is amazing, that thing that you said, that the peripheral blood, they have this regulation. I wonder if you have the ability to compare with the liver biopsies from some patients, because the biopsies, they are extremely limited or they are restricted to be obtained from patients. If you guys can be able to obtain some of that information. To compare.
No, that's one of the main limitations, that it is quite difficult to obtain liver biopsies unless you have a critical liver pathology that have the indication for a surgical intervention.